Welcome back to this next video, and in this video we are going to talk about the transforming growth factor beta 1 pathway via the receptor serine threonine kinase. So the first thing we need to understand is what are serine threonine kinases. When you talk about the kinase enzymes, kinases are those enzymes which add a phosphate group in its substrate protein on a specific amino acid. So when you talk about the serine threonine kinases are those kinases which add a phosphate group either to the serine amino acid or threonine amino acid in a substrate protein, thereby making the phosphoserines and phosphothreonine in their substrate protein. So serine threonine kinases have actually added a phosphate group to the serine and threonine amino acid in the substrate protein. When you talk about the serine threonine kinases, they are, you can say, one of the most abundant kinases in the living system, and at least 125 of the 500 plus human protein kinases, they belong to the family of the serine threonine kinases. Now these kinases, they switch between active and inactive state depending on the need of the body, and these serine threonine kinases can be activated by different mechanisms. For example, they can be activated after the binding of an activator, after the phosphorylation of their active sites, or after the dephosphorylation of the inhibitory phosphatases. So they can be actually activated by any of these mechanisms that we just discussed. If you look at the examples of the serine threonine kinases, the important examples are the receptor serine threonine kinase, which will be the focus of this particular video. Another important example of the serine threonine kinases, they are the protein kinase A, which is also known as the cyclic AMP dependent protein kinases. And another important example is the protein kinase C, which is also known as the calcium dependent protein kinase. So in this particular video, we are going to focus on the receptor serine threonine kinases and the transforming growth factor beta 1 pathway, and we will be focusing on the protein kinase A and the protein kinase C in the subsequent videos. So, if you look at the receptor serine threonine kinases, these receptor serine threonine kinases, as you can see, are actually transmembrane proteins. They have got an extracellular domain, they have got a transmembrane domain which is actually embedded in the cell membrane, and then you have got an intracellular domain. Now, the receptor serine threonine kinases, they are actually of two types. One is known as the receptor type 1 or the type 1 receptor, which are shown by these red bars over here. They are the type 1 receptor, and another type of the receptor, they are known as the type 2 receptor, indicated by these blue lines, by these blue bars over here. So if you look at the mammalian cells, there are 7 known type 1 receptors. So there are 7 types of the type 1 receptor, and there are 5 known types of type 2 receptors. Now, what happens is when these receptors are not involved in the signaling pathway, they are actually away from each other. If there is no signal for the initiation of the pathway, these receptors, the type 1 and the type 2 receptors, they are actually away from each other. When a signal comes, for example, if a signal comes in the form of the TGF beta, which is actually the transforming growth factor beta. This TGF is transforming growth factor, and then you have got this beta, so this is the transforming growth factor beta. When the transforming growth factor beta comes, it actually interacts with the receptor type 2 or the type 2 receptor. They do not interact with type 1 receptor, they actually interact with type 2 receptor. As you can see over here, when the transforming growth factor beta comes, it is actually binding to the type 2 receptors. When the transforming growth factor beta binds to the type 2 receptor, it actually recruits the type 1 receptor towards itself. As you can see in this particular image, they are away from each other, but after the binding of the transforming growth factor beta, the type 2 receptors and the type 1 receptor, they are coming close to each other, making a complex which is known as the heterotetrameric complex. Now this tetrameric means that there are 4 subunits involved, and you can see over here there are 2 type 1 receptor, there are 2 type 2 receptors, so total of 4 subunits they are there, therefore they are known as the tetrameric complex. As these receptors are different from each other, type 1 and the type 2 receptor are different from each other, therefore it is known as the heterotetrameric complex. Now if you look at the transforming growth factor beta 1, what is transforming growth factor beta 1? This TGF beta 1 is a multifunctional cytokine. It is a protein which is produced by all white blood cell lineages, and these transforming growth factor beta are involved in the cell growth, in the cell proliferation, the cell differentiation, and apoptosis. As you will see later on in the video, that these transforming growth factor beta are also the inhibitors of the cell cycle at the G1 phase of the cell cycle. So the transforming growth factor beta, as it is a multifunctional cytokine, it is performing a variety of functions in the cell. Now when the type 2 receptors and the type 1 receptor, they come close to each other in response to the binding of the transforming growth factor beta to receptor type 2, the type 2 receptor is going to phosphorylate the serine residues in the type 1 receptor. As we are talking about the receptor serine threonine kinase, that means that the type 2 receptor have kinase activity, and that specific kinase activity is serine threonine in nature. So when the type 2 receptor come in interaction with the type 1 receptor, it is utilizing its kinase domain to phosphorylate the serine residues of the type 1 receptor. So if you see over here, the binding of the transforming growth factor beta is actually making the receptor type 1 in the phosphorylated form, or it is generating phosphoserines in the receptor type 1. Before going to discuss the pathway, we need to understand the SMADs, because when this receptor type 1 it gets phosphorylated, it is going for the activation of a class of proteins which are known as the SMAD proteins. So before going into the pathway, we need to understand the classes of the SMADs and its phosphorylation. Now there are usually 3 types of the SMADs that are present in the living system. The first class of the SMADs, they are known as the R SMADs. They are the receptor regulated SMADs, and the members of the receptor regulated SMADs, or the R SMADs, they are SMAD1, SMAD2, SMAD3, SMAD5, SMAD8/9. Sometimes this is known as the SMAD8. In some literature you will find this as the SMAD9. So this SMAD8 and 9 that actually refer to a single protein. The second type of the SMAD, they are known as the common mediator SMADs, or for short they are known as the co-SMAD. And in the common mediator SMAD, it includes only a single member, which is known as the SMAD4, which is actually responsible for interaction with the R SMAD to participate in the signaling pathways. The last type of the SMADs, they are known as the antagonistic or inhibitory SMADs, and for short they are known as the I SMADs. This I is for the inhibitory. There are 2 members in this class. One is known as the SMAD6, and the other one that is known as the SMAD7. The SMAD7 that is actually responsible for blocking the activity of the R SMADs, and the SMAD6 they are actually responsible for blocking the activity of the co-SMAD, that is SMAD4. So these are the classes of the SMADs. Now when the receptor type 2, it phosphorylates the receptor type 1, what it do is that there is a protein that is embedded in the cell membrane, which is known as the SARA protein, and this SARA is the SMAD anchor for a receptor activation. This is the full name of the SARA protein. This is the SMAD anchor, and this is for receptor activation. So what this SARA protein do is that this SARA recruit the R SMADs, in this particular case the SMAD2 and 3 complex. This R SMAD actually refers to when you talk about the transforming growth factor beta pathway. This R SMADs are actually the SMAD2 and the SMAD3. So what the SARA do is that SARA recruits these R SMAD, this complex of the SMAD2 and the SMAD3, to the receptor type 1, and when the SARA recruits the R SMAD, it actually permits the binding of these R SMAD to the L45 region of the type 1 receptor. So this L45 region is actually a binding site for these R SMADs. What this SARA do is that it actually orient these R SMADs in a conformation that the serine residues which are present in the C terminus of the R SMAD that actually faces the kinase domain of the receptor type 1, and utilizing the kinase domain, the receptor type 1 is going to phosphorylate the serine residues in the R SMAD which are present in their C terminal. So when the receptor type 1 it phosphorylates the R SMAD, this is going to induce a conformational change in a specialized domain of the R SMAD which is known as the MH2 domain, and when there is conformational change in the MH2 domain of the R SMAD, it is dissociated from the receptor type 1 and from the SARA. Initially the R SMAD, they are in interaction with the SARA protein as well as with the receptor type 1. So when the receptor type 1 phosphorylates this, that phosphorylation is actually changing the conformation of this R SMAD, and in that changed conformation it is no more able to interact with the SARA in the receptor type 1, therefore it is dissociated from the receptor type 1 and the SARA. Now once you have got the phosphorylated R SMADs, these phosphorylated R SMAD are going to interact with the second class of the SMAD which are known as the co-SMAD, or the particular example is the SMAD4. So the phosphorylated R SMADs, they are going to interact with the SMAD4 making a complex, and this complex of the R SMAD and the co-SMAD like SMAD4, they are actually going to move from the cytoplasm into the nucleus of the cell. That means that these R SMAD, after its interaction with the SMAD4, this whole complex is moving from the cytoplasm into the nucleus of the cell, where it is going to change the gene expression of the cell. Now this transforming growth factor beta that actually cause the transcription of those particular genes which are involved in the process of the apoptosis. They can also activate the genes which are responsible for the extracellular matrix neogenesis. They are also activating the genes involved in the immunosuppression and block in those particular proteins which can actually arrest the cell cycle in the G1 phase. So as you can see over here, this is actually initiating a lot of pathways, and it is multifunctional in nature. So the if you can say summarize the whole pathway, the binding of the transforming growth factor beta actually leads to the phosphorylation of the R SMADs, and the interaction of the R SMAD with the SMAD4 is going to change the gene expression of the cell, thereby causing a cellular response. If you talk about the regulation of the receptor serine threonine kinase, how they are actually regulated. Now, as I've told you, one of the important class of the SMADs, they are known as the I SMADs or the inhibitory SMADs. In the class of the inhibitory SMADs, the SMAD7 that actually competes with other R SMADs like the SMAD1, SMAD2, SMAD3, the SMAD7 is going to compete with them for the type 1 receptor, thereby preventing the phosphorylation of the R SMADs. If you talk about the SMAD6, the SMAD6 actually binds with the SMAD4, thereby preventing the binding of the phosphorylated SMADs with the co-SMAD. Therefore, they are not allowing the movement of the R SMADs from the cytoplasm into the nucleus of the cell. So this is one way how this particular pathway or this particular receptor that is regulated. Another way that this particular pathway is regulated is by the ubiquitination of the R SMADs, and this ubiquitination actually leads to the destruction or the breakdown of the protein to which the ubiquitin molecules they are attached. I have a detailed video how the ubiquitin proteasome system works, and I'll link the share in the description. If you are interested, you should watch that video to get a better understanding of the process of ubiquitination and the breakdown of the proteins. Now, in the case of the RSMADs, there are specific E3 ubiquitin protein ligases, which are known as the SMURF1 and the SMURF2. They actually regulate the level of the RSMAD. What these SMURF1 and SMURF2, what they do is they transfer ubiquitin to the RSMADs, which cause their ubiquitination and their subsequent proteasomal degradation. Now, the SMURF1 that is specifically binding to the SMAD1 and the SMAD4, thereby causing their proteasomal degradation, and the SMURF2 that actually binds to again the SMAD1. Can bind to the SMAD2, the SMAD3, SMAD6 and SMAD7, thereby keeping their level within the required limits of the cells. If you like the video, please subscribe to my channel, hit the like button, hit the notification button, and share the video with your friends. It may be useful to them. So in the next video, we'll be talking about another important serine threonine kinase, that is the protein kinase A.